Understanding the nitty-gritty of how molecules interact with each other in the real, messy, dynamic environment of a living body is a challenge that must be overcome in order to understand a host of diseases, such as Alzheimer's. Until now, researchers could capture the motion of a single molecule, and they could capture its rotation, how it tumbles as it bumps into surrounding molecules, but only by compromising 3D resolution. The research was published in a special issue of the Journal of Physical Chemistry B. The February 17, 2022, Festschrift is dedicated to Nobel laureate William E. Lew's lab is the first to be able to visualize the orientation and direction of a molecule's rotational movement, how it spins and wobbles, while it's in a liquid system. The new imaging technology, called a vortex microscope, relies on a particular type of light, a polarized optical vortex. You can bend the light in a certain way so that the photons are spinning along their path, Lew said. Instead of a straight beam of light, this optical vortex is shaped more like a corkscrew. It's created by shining light through a helical-shaped lens, the top of which is uneven, sloping downward into a spiral. The microscope also splits the light into two different directions of polarization, providing insight into the direction of the wobble of nano-sized light sources, the molecules in the sample. For their experiments, Lew and first author Tianben Ding, then a postdoctoral researcher in Lew's lab, looked at amyloid beta fibers. Clumps of these proteins, found in the brain, are associated with Alzheimer's disease. The light carried information about its interaction with the fiber. After it passed through the lens, that information was translated by an algorithm developed by Lew's team. An optical vortex is not a point of light, but it's spread out in more of a donut shape. Based on the donut's properties, is it stretched out along a certain axis, or darker in some places? The algorithm can infer 7 distinct properties of the tracer molecule, including its position and direction. Because the team used a polarized optical vortex, they also can determine the direction of the wobble, a novel ability of the vortex microscope. The ways in which the molecule interacts with the fiber can, in turn, help paint a picture of the fiber's motion and topology. Putting it all together, the vortex microscope offers a detailed look into how the surfaces of these amyloid beta fibers interact with each other, how they bounce off each other or attach, and how their surfaces affect whether or not they begin to aggregate. This is the first time we can measure these very detailed dynamics of how molecules move and rotate inside liquid systems, Lew said. Tianben Ding, Matthew D. Lew. Science Daily, 17 February 2022. ift.tt/pso Rehay. Washington University in St. Louis. 2022, February 17. Vortex microscope sees more than ever before. In a first for imaging, new microscope captures details, 3D motion of molecules in liquid. https://ift. tt per pico Siemens 0 Rehay. Accessed February 17, 2022. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.